Today we'll be looking at muscle tissue. I try to repeat a lot, so hopefully this will be able to help. We know of the muscle or the muscle type or the types of muscle fibers that exist. The cardiac, skeletal, smooth. What's important here is the diagram on the right. This is just showing a basic comparison between smooth cardiac skeletal muscle. So it's basically showing the muscle fibers, whether they're striated, where the nucleus or the nuclei are usually located or situated. So for example, if you look here, this muscle look like what type of muscle? This here to the right.
The skeletal muscle.
Skeletal, perfect. Why do you think it looks like skeletal muscle?
It's arranging like a parallel arrangement, and also like it's striated.
Striated, one, good. What else?
There is an absence of the intercalated discs.
Yeah.
Found in the cardiac muscle.
And what about the nucleus? What's special about the nucleus versus cardiac versus smooth?
This is it, the second image on the right?
Yeah, the second image on the right. Yeah. Oh. This one. 1, 2, 3. Muscle fibers. Yeah.
They're pushed on the periphery of the muscle fiber.
Perfect. They're on the periphery. And if you compare that to the image above looks to be what type of muscle?
Cardiac.
Cardiac muscle. Why do you think it's cardiac muscle?
Honestly, I was just looking at matching it with the image on the left.
All right. I mean, you could look at also the labeling. So here we have intercalated disc. We were comparing it to this muscle down here. That is a skeletal muscle. And as Danielle pointed out, there's no intercalated disc. So the important thing about cardiac muscle is that there's intercalated disc. We'll go through that in a bit more detail. But if you also look at the nucleus is located more centrally sometimes, right? As you can see here. And if you look at the fibers, the arrangement of the fibers, look at the arrangement of these muscle fibers as opposed to the skeletal muscle fibers. And if you look down here, this is smooth muscle. No intercalated disc. Is there striation in smooth muscle? Are you seeing any? Are there?
No, miss.
No striation. What you can see is a central nucleus, and the cells look like a bit pointy at the edge. Nucleus central, completely different from skeletal muscle fiber, where you can have multiple nuclei along the periphery. There is striation, and if you compare that to the cardiac muscle has intercalated disc, a centrally located nucleus. Sometimes you can have more than one nuclei as well, and there's also striation. For the cardiac and the skeletal muscle, you basically have striations, but you don't have any for the smooth muscle. You remember anything about the intercalated disc?
Was it that it aids in like the conduction of the impulse?
Yes, and remember we're talking about things on the lateral surface of the cell that help with conduction, action potential, junctions, gap junctions.
There's a lot of gap junctions, yeah.
Gap junction, desmosomes. This is just a diagram that you don't even need to know. It's just putting this into perspective that there are a lot of skeletal muscles in your body. There are a lot of muscles in your body. Skeletal muscles. How many skeletal muscles do you think you have in your body? Random estimate.
120 is random.
What was that? I hear 3,000? Huh?
I said 127, not 3,000.
127? That's a pretty random number. What did Google say? How many skeletal muscles do you have in your body? And if you look at the picture, you don't have to know it. Look at the eyes, muscles, temporalis muscles, muscles for chewing, so many muscles attaching from how much? 650? Sounds about accurate. So you have a lot of muscles in your body. They all have different names. So you have 650 skeletal muscles approximately in your body. There are some people where they can have some familial disorders where they have missing muscle or an absent muscle, but on average you have a lot of muscles in your body. 600 and something skeletal muscles. What are skeletal muscles? They're muscles that attach to the skeleton. Your skull needs a lot of muscles to cover it. Your eyes, your nose, a lot of muscle attach from your neck, from the base of your skull, insert into the neck. A lot of them attach from the vertebra, insert into clavicle, this bone right here. You have a lot of them, especially here. You have a lot of muscle over your breasts. area, pectoralis major. They all have different names. You don't have to know these. I'm just putting it into perspective so that you can see that skeletal muscles, there are a lot of them. So when we talk about skeletal muscle, everything that you learn, you know that it's specific to 650 muscles in your body. If you look in your hand, biceps, triceps, all of this you probably already know. In terms of the embryologic origin of skeletal muscle, as we touched on before, skeletal muscles in general, on average, tend to come from mesoderm. This is just a diagram showing the embryologic development of skeletal muscle fibers. Here, they derive from the mesenchymal cells. The mesenchymal cells divide to produce what we call myoblasts. Remember, I said that the blast cells are the immature version of the cell. So what would the mature version of myoblast be?
Myocytes.
Myocytes, which is why we call the muscles myocytes, right? Cardiac myocyte, skeletal myocyte, muscle, even the fibers, muscle cells, myocyte. The blast cell is what, during the embryologic period, give rise to what we call myotube. So what these blast cells do, they form a syncytium where they attach to each other. And this is about around 4 weeks. 4 weeks in utero. A person who doesn't even have a belly, all of this going on in their body. Myotubes will eventually give rise to myofilament, and the myofilaments are what is contained in the myocyte. The muscle cell will have filaments or proteins, and these are what are primarily helps with contraction. You don't need to know the detail on this diagram. It's just putting it into a bit more perspective so that you can understand it a bit better. Just know that skeletal muscle comes from mesoderm and you should be fine. This is also trying to put it into perspective. This is the anterior surface of the body. It's like looking at the person from the front. You're looking at the front, anterior. There are a lot of muscles, skeletal muscles in your legs, your thigh, from your hip to your knee, from your knee to your ankle. They all have different names. Vastus, lateralis, all different names. You don't need to know. What you would need to know is the muscle cell itself. So you need to know the muscle fibers. You need to know that skeletal muscle, there's a thin sheet of connective tissue that wraps around the muscle itself. You know, the muscle insert into the tendon and the tendon go to the bone, right? So this is looking at the muscle from a bigger perspective. For example, you could say this is the deltoid muscle right here. This deltoid muscle is inserting somewhere into the bone, inserting from here to here. And where the muscle tissue itself insert into the bone, they use tendons. And tendons are connective tissue. If you look at the muscle, the entire muscle itself, muscle tissue, this is a skeletal muscle. We know it attaches to bone, and that's why it's called skeleton, because it attaches to the skeleton. For the entire muscle tissue itself, there's a thin sheet that wraps around it called the epimesium. So I might give you a diagram and you will need to know that there's the epimesium, and don't be alarmed, this is going to be repeated so many times. It might just be a bit longer than usual, hopefully not. I'm trying to do it as slow as possible so that you can understand it. Here you will also have another sheet of connective tissue. That's the outline here, and this is what wraps the fascicles. So these are fascicles. Fascicle 1, 2, 3, 4, 5 going around. These are fascicles. So around the fascicles you should have perimesium. So you have epimesium around the entire muscle tissue. Perimesium should be around the fascicle, and endomesium should be encasing the muscle fibers. Remember, I said something about sacroplasmic reticulum. Anybody remember that? Did I say that it was equivalent to something?
Endoplasmic reticulum.
Exactly, the endoplasmic reticulum. There is sacroplasmic reticulum in muscles equivalent to endoplasmic reticulum. What was the function of the endoplasmic reticulum?
Wasn't it like the site where you can have the processing of proteins for like the packaging and also for transport throughout the cell?
if you see sacroplasmic reticulum in a skeletal muscle or in the muscles in general, the sacroplasmic reticulum will contain calcium, iron to be released to aid in contraction of the muscle. They are what will activate action potential. The Sacral Lemma, remember we said that you have a cell membrane. The equivalent to a cell membrane in this case would be the Sacral Lemma. Here, we basically said this was a fascicle. Then you have the fibril, which is the fibers within the muscle. So you have fibers. They're simple, easy to remember. Thick and thin filaments. Thin and thick filaments. Thick filaments are the dark bands that you're seeing going across. If you look here, thick filament. This dark band going across. These are thick filaments. Do you know what thick filaments are made of? What do you think they are? It's a big machine. All machine needs something, and it's very common, and I use that word over and over. It starts with P, protein.
That's it. Okay.
It's not even complicated. It looks complicated, but it's just protein, right? So the thick filament, protein structures, thin filaments, protein structures, and you will eventually understand that these filaments are what will aid in contraction or muscle contraction. You always have mitochondria. They are specifically situated at the unique areas in the cell. And of course, you'll expect an abundance of mitochondria. Why exactly do you think you will have an abundance of mitochondria? What's the function of the mitochondria? We did this before.
To supply energy. Yeah.
ATP synthesis. The skeletal muscle is the most common muscle in your body. As you already know, it's 650 of them. It's voluntary because you decide when to move, when to run, when to punch. And that's different from the cardiac muscle that is involuntary. Functional unit of the skeletal muscle are the fibers. And we already talk about the muscle fibers. You just don't realize the fibers will contain the myofilaments. The myofilaments are going to be actin and myosin. Actin and myosin are contractile proteins. Sounds a bit complicated. No, you'll see all of this eventually, and hopefully you'll understand it a bit more than I did when I was trying to learn it. It's 40 to 50% of your body weight. Naturally, you have a lot of muscle, and it's attached to the skeleton, as you already know. They're striated and they're voluntary. It appears striated because of the cross bands that you see under the microscope. What do you think the striations are?
Due to the actin and the myosin.
The actin and myosin filaments, right?
Yes.
Exactly. The thin and the thick filaments are what the striations are. They're just protein in specific arrangement. So they're multinucleated, as we mentioned before. So if you look at this might just be a nice diagram for you. Look at this. There's nothing here. You can easily just drop this on a paper. So let's see what these things are. What do you think this is right here? These things.
The nucleus?
Nucleus. Nuclei, right? Multiple. We say it's multinucleated. So you're looking at multiple nuclei, and they're located on the periphery, as we already touched on. These are some dark bands, the striations that it's showing. What do you think the dark bands are? So you have thick and thin filament. Thick and thin filament. Which one do you think it would be?
The actin would be the thinner, I believe.
The thinner. So the thinner would probably be lighter, and the thick one would be darker.
Yes, miss.
Yeah, so you're probably looking at the filament, and this is a muscle fiber or a muscle cell. The cytoplasm is known as the sacroplasm. The endoplasm is known as the sacroplasmic reticulum, or the endoplasmic reticulum is known as the sacroplasmic reticulum. For muscles, when you see sacroplasm, cytoplasm, and you already know that cytoplasm, water, the organelles, protein. So it's packed with myofibrils, which we already said they're proteins, and they're cylindrical. The myofilaments are long fibers that are in the myofibrils. So the long fibers are the actin and myosin filament. Each myofibril has a unit and consists of identical repeating units called sacromeres. We'll go through the detail of sacromeres eventually. This is a sacromere. The sacromere is the unit where there is muscle contraction. So when you have muscle contraction, there are certain things that happen, and I think I'll pull up a video so you guys can see a bit more detail or understand it a bit better. Just remind me if I somehow get lost into this presentation. Here you can see at the bottom of the diagram, you can have thick filament. The thick filament we say were the myosin filament, thick filament, myosin filament, thin filament, actin filament. The entire sacromere is from between this line to this line, the Z line. So you have multiple sacromeres in our myofibril. Remember we said that these alternating dark and light areas are the actual proteins, the actin and myosin contractile protein. These will aid in muscle contraction. Between here, this Z line, and between here, this Z line is the sacromere. So what's between it? We already know you have a thick band. Look, I have the cursor right here. This is one thick band going across. You see it here? We're within the sacromere. We said this is the Z line. Z line here. We have a thick band going across. Thick, another one here, another one there, another one here. Just all these red thick bands that are going across myosin. And here you'll see eventually this is the M line right here. The M line is between here. If you look between this thick line in the middle, here you have actin filament thin, actin filament thin. This one here, this one here. They all meet the Z line right here. So you see when muscle contraction occur, I'm telling you everything now, but you know it already in the presentation, right? So when muscle contraction takes place, the actin will basically be pulled inward. So what contraction is actually shortening of the sacromere. Because remember that the Z line here, you have the actin filament along here attaching to the Z line. You can see it on both sides, right? So when contraction takes place, both at the Z line, just imagine the Z line will come together. So it's like the myosin will bind to the actin and it will pull the actin inward. In essence, muscle contraction is a shortening of the sacromere. Is when the actin filaments adhere to the myosin and is pulled towards the middle of the cell or the muscle fibril. This is not the best image, but we have a lot more. So I'm not even going to talk about the bands right here. We know that skeletal muscle make up a lot of your body weight, as we already said. The sacromere is the contractile unit because it contains the actin and the myosin filaments, and these are the contractile proteins in the muscle. We already know the muscle fibers insert into the tendon here, and it's a special type of connective tissue too, which you will see on histology. Right here is like the muscle tendon junction, and you can imagine what you would see in a muscle tendon junction: connective tissue, muscle fibers just going in. You'll see the muscle, the nucleus, and all of these things, and the connective tissues that are there. This is a clearer picture of the myosin. So the dark line that I was showing you on the other image, these are the myosin filament, myosin. Here, actin filaments. Remember we said both Z line here, Z line here. This is like the sacromere. Sacromere is a contractile unit, and when the muscle is contracted, it's because the myosin and the actin bind together, and what it does is pull the actin on both sides together. So it pulls both of them to the midline and shortens the muscle. I'm just introducing the concepts early so that when we go through it thoroughly, you'll be able to understand it a bit easier. So we're still on the external surface of the muscle. The muscle tissue itself is covered by a fibrous connective tissue. Within connective tissue, and you'll know that the perimesium, the epimesium, the endomesium, they're connective tissues, and these are fascia, or thin sheets. You remember the constituent of connective tissue? What do you expect to be in like the perimesium?
Oh, collagen.
Collagen, yes, synthesized by what
Brown
cell?
substance.
Ground substance, great. So what's in ground substance? What's ground substance?
Protein.
Protein. Remember any name? Protein of any kind?
I don't remember.
How about the GAG?
Glycosaminoglycan.
Exactly. Yeah, the proteoglycan. Hyaluronic acid.
Like, amino.
Acetylcholine, great. And they said fibers, right? What are the types of fibers we find in connective tissues again?
Elastic fibers.
Elastic, that's one. What else? Reticular fibers.
Reticular fibers.
And what else?
Collagen fibers.
Perfect. You guys doing well. Back to the muscle cell. We look at the entire tissue a while ago and we say it's wrapped by connective tissue, and the connective tissue is called the epimesium. So now we're actually on the muscle fiber itself, or the muscle cell. Remember that we said the muscle cell is surrounded by sacral lemma. Remember the membrane? This is basically like a cell membrane, the sacral lemma. It's difficult to remember, just remember 2 M's membrane. I don't know, maybe that works somehow. Sacroplasm is similar to cytoplasm, so you know that the plasm is the equivalent to the cytoplasm in the average cell. It's just that this cell is a bit different. It's long, it's elongated, it has a lot of proteins in it, but it still has the mitochondria, the Golgi, all of these things that you need to produce proteins, produce enzymes, to interact with ions. It does the same thing like all other cells, just that these cells are highly complex. They make you walk, they make you run. They're not no typical cell, right? So they are much longer. So the sacral lemma is a membrane, sacroplasm, cytoplasm. You have the mitochondria. So if you look between the myofibrils, the fibers, and these contain the actin myosin filament. The myofilaments are actin myosin filaments that are coming from here. You look at this typical cell, you know that it has its nucleus, just like all other cells. They're just located on the periphery, and they're just longer. You know that they have the mitochondria, and they're specifically located at nice location so that when the cell need the energy, ATP for contraction, it is there. And naturally, you're going to expect a lot of mitochondria in these cells because they're highly functional. You need the energy, you need to use the ATP, you need to use the glucose, you need to have the glycogen, depending on whether the cell is using aerobic, anaerobic respiration. However, the cell decides to use the energy, but naturally it makes sense that you'll need a lot of mitochondria. So just like the endoplasmic reticulum, you have the sacroplasmic reticulum, and this has very unique function in the skeletal muscle. As I said, it's there sitting, waiting for an action potential to signal when to release calcium ion. And I think I should do a lecture on action potential in like tissues in general. For example, the nervous tissues. I probably should separate the lecture where I do the action potential for skeletal muscle because it's pretty complicated. Well, it's not complicated, but it requires specific attention. I mean, I'll cover a bit of it, like how the action potential is carried out, whatever needs to happen, binding of receptor, releasing of sodium ion and calcium ion and all of that. But it requires a bit more focused attention to really understand it. The sacroplasmic reticulum will have a lot of calcium to release to help in muscle contraction. So as we said, the nuclei are elongated, and in skeletal muscle can be multiple nuclei. We know that they insert in tendons, and the tendons attach to the bone. So the muscle fibers insert into the tendon, as you can see here, and the tendon attaches to the bone. We already know surrounding the entire muscle tissue, there is a dense connective tissue sheet, epimesium. You guys already tell me all the fibers that you can find in connective tissues and specific cells, fibroblasts that helps with producing collagen. Perimesium is also a connective tissue, and they cover the fascicles. So remember that you have the big muscle, and then when you slice the muscle in half, you have these little round things in that you call the fascicle. These fascicles. Within, it's perimesium that covers the fascicle. Epimesium cover here, perimysium cover the fascicles, and the fascicles will contain the muscle fibers. The muscle fibers are the muscle cell. So you have one heap of muscle cell in this, and then you have the endomysium that covers the muscle fibers or the muscle cell. So epi means upon, so it would be the outermost. Peri, endo. Endo is always within, like the endothelial cells. Endoderm, within. This is just a histology picture showing a different perspective. Here you have the muscle fibrils, or this is the muscle fiber, and if you look between, you have the connective tissue. So the perimysium would be the lining of what?
Fascicle.
Fascicle, exactly. What would the endomysium be covering?
Muscle fiber.
Yeah. Here, if you look between, they're connective tissue. The periphery of the muscle, this is like a transverse section. So you can cut it transverse. You know what transverse is, right?
Yes, miss.
Yeah. So you can have a transverse view or a longitudinal view. So if you have a longitudinal view, you see the entire fibers, the long fibers, but now it's cutting off, so you're just seeing the half of it. So you're looking at it like you're looking at it from top. So you see the nuclei again in the periphery, but it goes down, all the way down, right? Here, nuclei on the periphery again. This is showing a bit more detail. Here we have the tendon. This is the muscle. So the epimesium would be on the outside. Then you have the perimysium, and then you have the endomysium that would be surrounding the muscle fibers. So perimysium, fascicles. So epimesium, the muscle tissue, perimysium. What's a perimysium again? Fascicle. And endomysium, fiber. Pull out one muscle fiber, because remember I said the muscle fiber is the cell, So you pull out one of the muscle fiber from the fascicle, pull it out. What you should see, the endomysium, you should also see the membrane. And what would be under the cell membrane? What would be the cell membrane?
The Sacral Lemma.
Yes, exactly. We know that the cell have a lot of fibers, and these fibers are proteins. They're actin and myosin filament. So if you look, actin and myosin filaments are going to be divided in groups of sacromeres. So this is a sacromere, even though it's not labeled. This is another one, this is another one, this is another one. And we already know that it is as a result of the rearrangement of actin and the myosin, myofilament. And we know the myosin is the thick one, that's the dark one that's usually in the middle. The actin is the light one that's on the periphery, where you add the Z line. This is a bigger look at the sacromere, the contractile unit. This is just looking at the same dark band that we were looking at. And we said the dark band was what? What filament?
Myosin.
Yeah, myosin filament. This one was the actin filament. There are different proteins also, like titin. You don't need to know that. Just know this part. Here would be the Z this, and we know already this is like the actin filament. See them? This purple. What you have over the actin filament are topomyosin, also another protein filament that goes around it. And then what you also have, this topomyosin locks the actin myofilament. Imagine if the muscle is not being contracted, these proteins are going to be like this, right? They're going to lock the actin myofilament in place so that the myosin doesn't bind to it. So these proteins that you see on top of the actin strands, see them here, the two purple things, they have some protein that wrap around it as well. Topomyosin. Topomyosin is the protein that covers the strands. I think these are phosphate groups as well on the strands. And what the topomyosin does, it creates that link between the myosin. So when the muscle contraction needs to occur and the actin needs to bind to the myosin, the topomyosin has to move or change in order to release an area, make an area on the actin myofilament and strand available for the myosin to bind to it in order for it to facilitate contraction. So we're still on the basics. What covers the muscle? This is just a histology slide of the muscle tissue. What do you think this area is or would be? So this is a transverse section, apparently, a transverse section of the skeletal muscle. And it's it skeletal because the nuclei are on the periphery. But look at this. What is this right here? What do you think it is?
That looks like
It's a connective tissue.
connective tissue. Yeah, I was thinking.
Well, what is the connective tissue E? More likely epimesium. Epimesium.
Yeah, epimesium. The surrounds it.
Exactly. So what would these be? What do you think the entire thing is?
A fascicle.
A fascicle. So what do you think would be around the fascicle?
Perimesium.
Exactly. And what do you think this would be? What do you think these are? So one nuclei. These are the muscle fibers, right?
Yeah.
Muscle cells, individual cell, one cell, two cell, whole heap of cell make up a fascicle. So what do you think this would be? So wrapped around the fibers.
Endomesium.
Endomesium, perfect. So you see, you are able to identify it on a histology slide. What you can do when you come next week, when you guys come next week for the session, try to identify these things. That's the best way you can learn. And trust me, you'll remember it probably for a long time if you learn it that way. All right, this is just another diagram showing the perimesium. So the epimesium is lacking. So the epimesium would be here, perimesium around the fascicle, endomesium around the fiber. And this is just showing the rearrangement of the organelles in the muscle cell. So the organelle, we know the outermost layer is the endomesium, and that obviously on the inside of that you would have the sacral lemma, because that's the plasma membrane. You will have the nuclei, they are peripherally located, and you'll have the mitochondria. You need a lot of them for ATP. And of course, you have a lot of muscle fibers or myofibrils, and these myofibrils will give you the actin and myosin filament. You also have the sacroplasm. Don't forget the cytoplasm. This is a specialized type of cell that is present on skeletal muscle that is not present in the heart muscle. And this cell is for regeneration. It's like a fibroblast. When the cell is damaged, this satellite cell that is usually on the periphery of the muscle fiber or cell itself, this basically helps in regeneration. So it's going to produce whatever in order to regenerate, and the proteins in order to regenerate the cell. Because naturally you get injury, right? You get injured. Obviously, there has to be some intelligent design, which is the satellite cells that will help you to rebuild. The disadvantage, though, these satellite cells are not present in your heart. So you understand why when people get heart damage from high blood pressure, from heart attack, they have permanent damage because the cardiac myocytes don't regenerate like the skeletal muscle cells. When you get heart damage, it's usually permanent. The cells recycle once, by the time I think you're 40, you'll no longer be regenerating any cardiac myocytes, and the regeneration takes long. So basically, when you have any chronic problem, or if you have something that was chronic, like high blood pressure, diabetes, that can damage the heart. When you damage the heart, it's permanently damaged, because most people by age 40, age 50 by having high blood pressure will cause damage to their heart cells if they're not controlling the blood pressure. And there's the physiology behind it, which I obviously won't go into. These patients, when the cardiac cells are under stress and they have to remodel, there's no satellite cells to help to reproduce these cells. So they end up with permanent injury. So sometimes the cells, the fibers become lengthened. So the muscle tissue itself becomes dilated. And if the heart is dilated, imagine when it's pumping, it can't pump the blood out of the vessels or through the lumen or the vessels properly. So that's why some people end up with heart problems, and the heart problems usually cause their demise most often because the cells for regeneration are not present. This is just another picture of the layers as well. Sacral Lemma, we know, is the plasma membrane. The vascular supply comes through capillaries. These fibers need blood. They need nutrients. They need everything. So it's the blood that will carry the nutrients, the ions, whatever it is that needs to be discharged within the cell. They're going to be between sometimes the fascicles. Each muscle fiber is invested by a thin external lamina, but the important point is that they're enclosed by a satellite cells that are closely adhering to the sacral lemma. So the satellite cell just itch on to the sacral lemma, or the membrane of the cell. And you already know the contractile units, myofilaments, actin, and myosin.
What is the means of transfer of the materials from the satellite cell to the muscle fibers?
The means means of transfer? What do you mean? As in when it's synthesized, how it's transferred to the cell?
Yeah, does it go through like gap junctions or does it use like packaging like exocytosis and so on?
More than likely, they will synthesize proteins, well, different types of protein. Not sure. Well, when proteins are synthesized, they have to be processed and packaged. But in terms of release, more than likely, they have like stem cell properties. The stem cell properties would mean that they can regenerate or recreate muscle fiber. But in terms of the mechanism,
Thank you.
yeah. So yeah, the Sacral Lemma is glycoprotein. It's like a cell membrane, as we know, and it's just glycoprotein, and it's a network of reticular fibers. They have reticular fibers, and not surprised because there's connective tissue overlying. What's important about the Sacral Lemma or the plasma membrane, it invaginates. You know what invaginate is, right? Like it goes inwards, grows into like a tubular structure that goes inside of the cell. So that the invaginate, you call it a T-tubule, because when it invaginates like down into the cell, you see right here, it forms like a T. So it's just the Sacral Lemma that is dipping within the cell that's forming this T-tubule. This T-tubule, you'll eventually realize, is very important when the muscle needs to be contracted, because the Sacral Lemma will get the response from a receptor, because remember the Sacral Lemma is a membrane, right? And you remember that the membrane, cell membrane, have all the things on it, like receptor, ion channel, and all of these things. Sacral Lemma is going to have receptors. So when the action potential is generated, for example, there's a receptor called Acetylcholine Receptor at the nerve and muscle junction, where the nerve meets the muscle. The nerve terminal, let me not say it without thinking. The nerve terminal will release vesicles of acetylcholine, and this will bind to a receptor. When it binds to the receptor on the Sacral Lemma, it will result in the influx of sodium within the T-tubule. So it's like the sodium goes into the T-tubule, that invagination, and when it goes into it, it basically stimulates the action potential, where the Sacroplasmic Reticulum, this one down here, will release the calcium. And the calcium is what will eventually go and interact with different parts of the actin at the myosin, and the myosin will basically bind to the actin and it pulls it inwards, and that's how you get the contraction. You don't need to know the length or the diameter of the myofibrils, but sometimes just remembering random numbers like 1 to 2 micrometer, it's putting it into perspective and dimension. I randomly usually remember like specific things like these for most things. I don't know why, but I think it also helps to understand the bigger picture, because when you understand micrometer, you understand millimeters, milliliters, you have an idea as to how to quantify or see it in a different way. So we know the myofibrils are the array of the filaments. The sacroplasmic reticulum, we say, similar to the smooth endoplasmic reticulum. It is elongated and they're tubules. So they're tubules. If you look at it, tubules. And in the sarcoplasm, you have ribosome, glycogen, lipids. Remember, we talk about these as, well, not the ribosome, but the glycogen and the lipid as inclusion bodies that are just there. For example, when there's a need for energy, you can have glycogen being broken down. You can have the fat also being broken down, reversing to give you the carbohydrates so that you can get the energy. The sacroplasmic reticulum is a special type of smooth ER, and it is found in striated muscle. What are the striated muscles again?
Skeletal and cardiac.
Skeletal and cardiac. They expect the actin and myosin filaments to be in both of them. So the sacrotubular system is just the T system. Remember, I talk about the T-tubule, which is an invagination of the Sacral Lemma, where the sodium can go down and trigger the sacroplasmic reticulum to release the calcium so that you can have contraction. Yeah, it's a triad system. I'll tell you that in a bit. So we already know that the mitochondria mitochondria are closely associated with the myofibrils because this is like a machine. It's an energy machine. You need energy. Can't fully emphasize that mitochondria is just going to be a very important part of it. So the sacrotubular system comprises of the T-tubule, as we say, and the sacroplasmic reticulum. And you know the T-tubule is an invagination of the Sacral Lemma, and the sacroplasmic reticulum is basically equivalent to the endoplasmic reticulum that stores the calcium that is very critical in excitation, contraction, coupling, and it basically means exciting the cell and facilitating contraction. So as we said, the sacroplasmic reticulum it's very important in releasing the intracellular calcium. Calcium within the cell, it's tubules, so they're just arrays of tubules and they have calcium in it. So if you look at the diagram here, this is a better way of understanding the sacrotubular system. This is the membrane, the Sacral Lemma, as we said, 2M membrane, plasm, the cytoplasm, and we know the similar to the regular cells, endoplasmic reticulum, sacroplasmic reticulum in this case, and it's functioned to house the calcium that is going to be used for contraction. There are also receptors that are necessary, and as you know, the cell membrane will have the receptors or will interact somehow with receptor to create actions in the cell. So here they have this triad. This includes the terminal cisternae and the T-tubule. They basically call this the triad. Helps to produce the sacrotubular system and the sacroplasmic reticulum as well. This is the T-tubule. The only thing that's missing from this picture that will help you to understand how it creates action potential is the neuromuscular junction. So here you will have the synapse, the nerve terminal would be right here, touching the membrane, the Sacral Lemma, and you'll see that on a diagram eventually. The nerve obviously have to touch the muscle in order for the signal to come from your spinal cord, wherever in your body, to create you to move. The nerve has to be contacted with or connected with the cell. So when it gets to the membrane of the cell, it releases the acetylcholine receptor that binds to the receptor on the membrane, and then it causes sodium to come in the T-tubule and then interact here with the sacroplasmic reticulum that will release the calcium that is necessary for action potential. And all of this is ATPase dependent. All of this is ATP dependent, which is why I have a lot of mitochondria. Yeah.
So what I see that he has a T-tubule and a terminal cisternae. So is it that there are two cisternae to make it a triad?
Yeah.
Oh.
So as we said, you have the channels, voltage-gated dihydropyridine receptor or dihydropyridine receptor on the T-tubule that is also connected to a ryanidine receptor. This is the calcium receptor. This one here, this is the ryanidine receptor on the sacroplasmic reticulum, interacting also with the dihydropyrimidine receptor here. You don't have to remember that. You just know already that there's receptor on cell membrane. The sacrotubular system, there is elongated mitochondria, pleomorphic mitochondria. In terms of the mechanism of contraction, sometimes you can have aerobic and anaerobic respiration or ways in which you produce ATP. So you have different ways, it can be aerobic, anaerobic. As long as you get the ATP for muscle contraction, that's what you care about, right? This is a high resolution electron micrograph that is also showing the sacromere. So remember, we said the sacromere is the entire contractile unit, and you have the sacroplasmic reticulum here. This is supposed to be a mitochondria. You have mitochondria that's supposed to be on the periphery sometimes, and it's always in location or in relation to the sacromere. Because remember, the sacromere is a contractile unit with the actin and the myosin, and the mitochondria, the ATP will need to bind to the myosin filament. Here you have the Z lines, you see them here, and here would be the M line, and we know the lighter areas would be the actin, darker area myosin, and the contraction will pull the muscle or the filaments together or to the midline. So this is a bit different, where you have the nerve attaching, the motor neuron attaching to the muscle. Remember that image? All I said that was lacking, the motor neuron attaching to the muscle. Motor means it's for movement, right? So the motor neuron will attach to the muscle. This is the Sacral Lemma, the membrane. This is the cytoplasm, sarcoplasm. This is Sacroplasmic Reticulum here. Remember we said that they're tubules. They're just tubules with calcium. And you should see the transverse tubules, which are the T tubules that are an invagination of the Sacral Lemma. So these tubules, the sodium will go into this and stimulate these. So they're actually even overlapping. And these are just the arrangement of the actin and the myosin filament. This is like the entire muscle. Not the entire muscle, but the fiber. These are just the filaments arranged. And when there's action potential generated, it comes to the synaptic cleft, bind to the receptor on the Sacral Lemma, the membrane, and then the sodium will go into the T tubule. Because obviously the T tubules are going to also be around here because it's an invagination. So the sacromere is a contractile unit, as we discussed before. There's the Z line. You have two of them. You have the Z line. This is just the histology. The sacromere is the entire unit here. This is the sacromere electron micrograph at the top. Remember, we say you have the two lines here. The entire sacromere unit is the contractile unit, and you have many of them. So if this was showing the length of the muscle fiber, you'll have another sacromere here, another sacromere here. There's just a lot of them organizing to pull on the fiber, pull the fiber to the middle. So this is an electron micrograph. This is a histology. So there are different ways you can see these things on the cells. If you're deprived of equipment, obviously, you're going to be at a disadvantage. Between here, the Z line, and here, you have the I band. This is a poor diagram to explain that. So I'll just explain this when I get to the next diagram. The contractile unit consists of the Z line, the A band, the H zone, the I band, the M line. I would say know this because this is the most important thing about skeletal muscle. The contractile unit, cardiac muscle, the contractile unit. So let's see if you can identify this on a histology. So when you come in the lab, you can look at skeletal muscle, and when you see the striation, you can at least feel a little bit more confident that you can have an idea as to what's going on there. In this area, or the dark area, because there are different ones, right? Dark area, you have the M zone. The dark area here, you already know, myosin filament. The light areas, actin filament. So you have two I bands, as you can see, the sacromere, two I bands here, one here. You have the Z line. This is the outline of the sacromere, the Z line, the Z line. The thin filaments are going to be overlapping where the I band is. If you look here, thin filament, Z line to the external part, thin filament, thin filament. So two I bands, thin filaments. M band is basically usually in the middle where the myosin filament is. There's also a H zone. As you can imagine, when it's relaxed, the sacromere is going to be a bit longer, because remember we said that contraction is when it gets shorter, it pulls the fiber to the middle. The sacromeres are going to be longer, around 2.5 micrometer when they're not contracted. These bands, as you can imagine, when contraction takes place, remember it's shortening at the sacromere, so these lines, the Z line will come together.
Excuse me, miss.
Yeah?
Why does the Z line stain so deeply, or why is it so dark?
Z line stain deeply.
Yeah, why is it so dark in color, the Z line?
Because there is the myofic filament. So I assume that the area where you have more proteins or thicker proteins tend to be a bit more dense. Or if it's histology, it will trap a bit more of the stain.
I know that the myosin causes a dark band like in the middle.
In the middle.
So, and then you have the, like I see where you have on the edges, like it's kind of light because of the actin. But then the Z line now, is there another protein there or?
Okay, so yeah, as I said, remember I said there are other proteins, you don't really need to know them. There are other proteins that are also associated with it, but they're not really like the core part of the contractile unit. So the chitin actually connects the Z band to the M band. And remember we said the M band is here. So from here to here, yeah, I think that should Could be. Yeah, I never really looked into all of those other proteins because I mean the important ones are the contractile ones. There are others that if they're affected, yes, like even the titin, if they're affected, they can produce disease as well. But I guess the primary thing is for you guys to understand it where the contraction is concerned so you can relate it somewhat to mechanics. I would think that would be a more logical way of looking at it. All right, so here, let's see if this is showing anything. Most of these diagrams, guys, they're showing basically the same thing, just a little bit more detail or a different perspective. So for example, this is showing more the T-tubule. You can see the T-tubule invaginating, and you can actually see more of the Sacroplasmic Reticulum invaginating. So you actually see that the T-tubule run along or even parallel to it sometimes. And it makes sense that if there's the release of whatever down the T-tubule, the Sacroplasmic Reticulum are literally adjacent to them, and instantly you can coordinate voluntary movement almost instantaneously. This is also just showing a different perspective. Z-band, I-band, tubules, and here the A-band we know is the myosin region right here. Yeah, and I mean you already know that there's lipid and glycogen and all of that in it. All right, so yeah, the intercontractile unit, again the sacromere, 2Z, and then you have the I band here. So this is just a histology and an electron micrograph showing the same thing, showing the nucleus at the periphery, showing the striation as well, showing the mitochondria. Yeah, there's nothing special about it. It's just the mitochondria, which we expect to be here, the nucleus, which we expect to be here, and yeah, Z bands as well. It's also a transverse section, and you can see the nerve here contacting the muscle fiber. Histology, muscle nuclei at the periphery. Another one, nuclei at the periphery, mitochondria, and you know the myofilaments are the protein structures that are within it. And yeah, this is also a nerve as well. So the neuromuscular junction is the nerve muscle junction, and that's where the action potential will be initiated. All right, so this is a bit more detailed diagram on the sacromere itself. There are parallel arrays of thin filament that span the I band. I think I have this picture a bit bigger. So you have parallel arrays of thin filament that form the I band. So if you look here, between here and here, you have the I band. So if you look up here, the Z is basically the entire sacromere. They demarcate the entire sacromere. So if the Z is the equivalent right here, is a thin filament projecting in, thin filament going to the next area. So the Z, this area, as they said, Daniel, the proteins that are anchoring this thin filament from here to here, thin filament from here to here. Because remember, it goes in and it goes out because the sacromere, another sacromere is going to be beside it. It's going to be attaching. The Z area is going to also be where the thin filament attached to or interacts here with a thick filament. So thick filament, thick filament, thick filament, thin filament, thin filament, thin filament coming down, and then you have the Z band, and then you have the thin filament going across here again. So yeah, it also overlaps the thick filament, as we say here. The third filament system is made of a single molecule titin, one of the largest known protein that connects the Z band to the M band. So the Z band is connected to the M by the titin protein. So that actually answers the question or confirms the answer to the question. The titin protein is going to be here, and it will attach the right here thin filament with thick filament. The thick and the thin filament will need to bind together, and then it pulls everything to the middle in order for contraction to take place. This titin contains elastic element that acts as a molecular spring and contribute to the passive elasticity of the muscle. So the titin, it's very important because it makes it elastic so that when the contraction takes place, it can allow it to recoil, can shorten and recoil. Nebulin is another protein that spans the length of the filament and forms the fourth filament in the skeletal system. You don't have to know these proteins as long as you understand the sacromere, the contractile unit, and the areas on it. That should be fine.
Okay, so you're saying we don't necessarily need to know about the titin and the nebulin?
No, I mean, you have it already in your memory. At least you already know it. So I'm not going to stress you to say, oh, no, Nebulin Titan, what they do, whatever. The most important thing is the contractile unit, the actin and the myosin filament. And once you know those exist, if you even go further research or continue researching, you'll have to read so much that you'll eventually know that titin, nebulin, and all of these things, even genes and enzymes and proteins that you didn't even hear from me, you will know that exists. But for now, it's just primary exposure. You don't have to know all of that. So, you need to know that this area is the thick filament. This area is the thin filament. This is the Z-band. The Z-band is basically at both ends of the sacromere. And you know that there's a protein that attach this thin filament to this overlapping thick filament. So the entire area where the thick filament is the M band. This between the end of the thick filament and the other thick filament is the I band, and you see it on either side. This is a relaxed muscle. But when there's contraction, if you look at this diagram at the top and you look at this diagram at the bottom, you see the big difference?
Yes, Miss, there's more overlap.
This diagram over here is showing a little bit more detail as to the contraction. You have the thin filament, which is the actin. And remember, we talk about the troponin. There are protein, tropomyosin. Tropomyosin encases the protein molecule itself. You also have troponin that encases the molecule. So this myosin, and this is the myosin, and this is the myosin. The myosin head will have to bind to an area on the thin filament or the actin. And in order for that to happen, this topomyosin will have to be removed in order for the myosin head. And this requires ATP. That's what this is saying. The myosin head will bind now to the myosin binding region on the actin, and then it pulls it. So a little bit more up close look on the myosin structure is a protein molecules. It has an actin binding site. We know it has to bind to the actin in order for muscle contraction to take place. And it has an ATP binding myocyte. We know that ATP is necessary for it to bind to the actin. This is just a bigger diagram. Everything we have a head, have a tail apparently. Yeah, it has a flexible hinge region because remember it has to move up the head. This is just showing the actin contractile unit, the myosin contractile unit. The actin here, we already touched on this, but here you have the topomyosin. The topomyosin is what locks the actin. Remember, you're not always having contraction, so this topomyosin is going to be there to lock or cover the myosin binding site if the contraction is not taking place. This is the confirmation that it will hold. It will have the topomyosin covering the myosin binding region and the troponin as well. So the troponin will lock the tropomyosin onto the actin filament when there's no contraction. We already know the actin filament has two proteins, tropomyosin and troponin. These proteins are also present in the cardiac muscle. So when you have muscle injury, sometimes it releases these proteins. For example, if you have a heart attack, it will release some of these proteins. Well, there's an objective way of measuring if a person has an acute cardiovascular event by checking for these proteins that are supposed to be locking over the actin filament. So the troponin complex responds to calcium ion concentration. So it is when the calcium ion is released from the sacroplasmic reticulum, it binds to the troponin. So when it binds to the troponin, it causes the removal of the tropomyosin so that the myosin can bind to it. So the myosin heads bind to actin and draw the thin filament a short distance past the thick filament. Then linkage break and reform farther along the thin filament to repeat the process. Filaments are thus pulled past each other. So another perspective, you already know the tendon, the muscle tissue, the epimesium, the perimesium surrounding the fascicle, the endomesium surrounding the fibers, and then the fibers consist of the myofilaments, the myofibrils, which contains the myofilaments, and the myofilaments are the actin and myosin, and the actin and the myosin form sacromeres that are the contractile unit, and these sacromeres are demarcated demarcated by the Z line, thin filament, thick filament. You guys should know this. Should be an expert at this by now. So the myofilament is an electron-dense cross-section. Will show the thick filament to be more dense because the filament is thick. It will be twice as much as the thin filament because it's thicker than the thin filament. The I band show only thin filament. The A band shows thick and thin filament, which appear as a hexagonal network of myosin fixed at the M band. 8th zone is the central segment of the thick filament that lacks cross bridge. So this is just describing what you would see or why you see what you see on an electron microscope. So you have different types of skeletal muscle fibers: type 1, type 2, type 2A, type 2B. You don't have to know the details about the fibers. Just know that they're different. They're categorized differently because the contraction is different. So some muscle fibers are for rapid contraction, some are for slow contraction, some use aerobic versus anaerobic respiration. It's just putting it into different perspectives that not all muscle fibers will be the same or operate the same, even though they're all skeletal muscle. But the type 1 muscle fiber are known as the red muscle fiber. They're aerobic. They're small. They have a lot of mitochondria, and naturally, because they're small, they're aerobic, but they're slowly contracting. There's low glycogen and there's low ATPase activity. So we're just looking at the fuel, the things that produce the energy for these contractile units. So mitochondria, the glycogen, the ATP, the fatty acids, all of these things are going to be recurrent terms in your mind when you're thinking about energy, when you're thinking about action potential. The type B muscle fibers are the white one, type 1 are the red one, type B are the white one. These are aerobic. They're very sensitive to fatigue. They have less mitochondria. They will differ based on the composition of mitochondria, whether they have glycogen, and because of the need for ATPase and oxidative phosphorylation. So depending on the characteristic of the muscle fibers, the ATP, availability of ATP will be a significant factor. The body is intelligent by design. If you understand the foundation, and things will be easier to understand. We only categorize things because there are differences, but the important thing is to understand the foundation in order to understand the bigger picture. All right, so the muscle tendon junction is where the muscle fibers meet the tendon, and we know that the tendon inserts into the bone. We have histology of muscle tendon junction. When you come in, you can look. You can see the muscle here, the skeletal muscle with the striations, and you see a cell here, and this the tendon. So this looks like the tendon. What kind of cell do you think this would be?
Fibroblast.
Fibroblast.
To secrete the collagen.
Yeah, exactly. So there are membrane proteins that are associated with the muscle tendon junction. We already know that there is extracellular matrix and with fibers that will connect to the membrane, the Sacral Lemma, the membrane of the cell. You will have different protein that will connect the cell to the extracellular matrix. Vinculin, they're also connecting with the internal muscle protein structures like the actin myosin. So the actin myosin filaments are within the cell, and obviously you have different proteins that are connecting to different proteins. And remember, we already know that integrin is a membrane protein. This is connecting to integrin. And then you have proteins in the extracellular matrix. They're also connecting to proteins. So you can have the collagen fibers, reticular fibers that these can connect to. As long as you understand that protein, that's where the science is now. Protein, genes, enzymes, protein, genes, and enzymes. This is a bit more information as it relates to muscle contraction, what happens at the nerve terminal or at the neuromuscular junction. Neuro mean nerve, muscle junction, at the junction where the nerve meets the muscle. Remember, it's the nerve that has to stimulate the muscle to contract. You do it voluntarily from your brain, but you're initiating all of these chemical reactions using your brain voluntarily. Imagine that. So you realize the power of the brain is also far more than we think.
Yes, miss.
So if you think that you want to move, what will happen? Your intelligent brain will send a program to this neuromuscular junction, to these nerve terminals, and what it will do is triggers it to release acetylcholine, and the acetylcholine will be released in vesicles that will bind to acetylcholine receptors. Bind to acetylcholine receptors will open sodium channels. You already know that there are receptors and channels on the cell, so it will bind to these receptors and open the sodium channel so that sodium ion can go in. Remember that sodium is usually in the extracellular compartment. Well, I never said it before. There's a balance between sodium, potassium, and these ions in the intercellular versus the extracellular compartment. So the sodium will be released into the cell, activate the action potential. The sodium goes into the T-tubule and stimulate the release of calcium from the Sacroplasmic Reticulum. Remember, when the Sacroplasmic Reticulum releases the calcium, it will bind to the troponin, and the troponin will get the signal to remove thyself. Tropomyosin will say, all right, you move, let me move, so that the head of this filament can bind in order for you to facilitate contraction. This is just, it looks like an electron micrograph of the nerve terminal. You have the receptors on the cell. You have the vesicles, the synaptic vesicles that are released, stimulate the receptor. And this is the voltage gated channel in the Sacral Lemma. The voltage gated channel will tell it to release the sodium, and it will go into the T-tubule, which eventually influence the Sacromere. This is a more chaotic view of the structure, but it's also very clear. The clinical point for skeletal muscle, cardiac muscle should be a bit less. The muscles are very complex, so the lectures, you just have to expect it to be probably a bit longer than the average. You have a disease or a condition called Duchenne muscular dystrophy, and I'm telling you all of these clinical points that you can research, So if you decide, say you want to come to one of these fancy research with me, no problem. If you decide that you want to go abroad and you want me to help you formulate a proposal or something to look at some mutation in skeletal muscle, if you want to engineer using cellulose or whatever, if you want to engineer some fibers, if you want to engineer some collagen, you should be able to do it as engineers, because that's the point of medical engineering. You guys should be knocking out genes, creating proteins, synthesizing proteins in the lab, having lab models where you knock out genes and use different types of protein to influence gene functions. You guys should be at that level. There is a protein called dystrophin that is a very important protein that helps to stabilize the actin. The protein is usually anchoring the actin. In Duchenne muscular dystrophy, there is no dystrophin. So there is the dystrophin gene that is usually on the short arm of chromosome 21. There's a mutation and there's no dystrophin to stabilize the actin. So you have a very unstable muscle, and an unstable muscle means that it's a weak muscle, because if the actin is not stabilized, remember, it's the actin filament that are pulled or shortened for contraction to take place. So imagine these people have weak muscle and can't really initiate no muscle contraction. So as we can see here, dystrophin maintains the mechanical integrity of the cell during contraction. So while this actin is being pulled, dystrophin helps to maintain the mechanical integrity. In mechanics, you guys should be looking at a way in which you can create an upregulation of dystrophin, if that's possible, or create a structure that could stabilize the actin filament, similar to a dystrophin protein molecule. I don't think it's difficult. I think it's more like you needing somebody to show you the relevant areas where to look. I believe strongly that the geniuses are here. I see a lot of positive energy, a lot of energy, negative energy even in Jamaicans. And the flip side to it, I think the energy sometimes is power, whether it's negative or positive. It's just a matter of redirecting that energy or refocusing the energy into the direction that it needs to be. A lot of people are just lost lost and distracted. So if we can program a lot of young men to start looking at the cells, the muscle, looking at the genes to see if they can fix for this problem. This is a problem of young kids. You're born with this genetic mutation. Is it that you can search, for example, in a mouse model where you can knock out the dystrophin, which I'm sure they've done, to see what alternatives there are? But you'll be surprised if you start reading article on the mutation and what people have done so far to figure out how to prevent exacerbation of the disease. You'll be surprised. There's a lot of things out there in the literature. This is what it should look like. The actin filament here, the dystrophin is supposed to be stabilizing the actin filament while there is a muscle contraction. The actin filament is not stabilized. So as a result, there is dissociated dystrophin protein complex. So these people with a mutation on the chromosome, they end up having kids who they are born and there are no symptoms when they're born. But you see, when they reach 5 and them growing and the skeletal muscles keep extending and growth factors keep influencing the growth, then you realize that the muscles become weakened. By the time they reach a certain age, they're wheelchair bound because the muscles are so weak. They tend to die pretty young. This is showing the satellite cell on muscle fiber, electron micrograph of skeletal muscle. You have the nucleus here, the big nucleus. You have a lot of free ribosomes in the cytoplasm as expected. Smooth endoplasmic reticulum, rough endoplasmic reticulum are going to be in this. Remember, the satellite cell is not the muscle cell or the muscle fiber. You have diseases that can affect the neuromuscular junction. It not necessarily affect the muscle fibers or the muscle itself. The problem can be at the neuromuscular junction where the nerve meets the muscle. Remember that the action potential has to come from the nerve terminal through the vesicle stimulated acetylcholine receptor on the cell. Remember, it doesn't come from the muscle, it comes from the nerve. So if you have a problem at the neuromuscular junction, which you tend to have in a condition called myasthenia gravis, then they can have weakness. So sometimes what they have, impairment in the acetylcholine receptor site, there's no muscle contraction, and they end up with what we call ptosis. So the eyelid just drop down. This is just showing the axon terminal, the nerve terminal, synaptic vesicle, the synaptic cleft. So there's also a condition called rhabdomyosarcoma. This is basically a tumor of the muscle and it affects kids sometimes. The tumor sometimes can arise from the satellite cell or the mesenchymal cell, and they are usually striated muscle. So there's a mutation again, genetic mutation, where the tumor cells express even antibodies to the transcription factors that are regulatory for the muscle itself. So like PAC, these are genes. You can look for these genes to see if there's mutation, for example, in patients with rhabdomyosarcoma. And if you look at the histology slide of a patient with rhabdomyosarcoma, you know what it should look like over here, especially with the cells. So you know that the nuclei are going to be enlarged, there's going to be disarrangement. All right, so the cardiac muscle is different from the skeletal muscle. Yes, but we know that they have similarities. One, they're both striated. Two, they also have the actin, the myofibrils, the actin myosin filament, and they have the striation. The difference is these are involuntary. They're involuntary, and cells are known as myocytes. They're branched, right? So they're not tubular and long. They're branched. They are joined by intercalated discs. The nuclei are oval. Sometimes you can have two nuclei, so it can be binucleated. The nucleus, if it's there, sometimes it's in the middle. So it's shaped a bit different from the skeletal muscle. And if you look at the histology slide up here, this right here would be an intercalated disc. So this would be one cell over here. This would be one cell over here. So this is the muscle fiber. So you can have the nuclei or the nucleus in the center. Sometimes you can have more than one nuclei. This is just a capillary. So sometimes when you see a little confusing stuff in the slide, I never really showed you guys like blood vessels and nerves, but if you see like these little things and the red, these are capillaries. So sometimes you can see like a blood vessel with blood. See this? Like a big blood vessel, food muscles.
When we were looking at the section of the lip yesterday,
Yeah.
I think we saw a blood vessel in like the glandular area.
Nice. So like this, you know, this is a blood vessel, and this is like a smooth muscle, and there is endothelial cells. So the epithelium would be a simple epithelium with endothelial cells just going around. And you know, the fibers sometimes should be elastic fibers because the blood vessels are supposed to be stretching or allowing recoil. They look way different from the skeletal muscle. As in the nuclei, the skeletal muscles are obviously pushed to the periphery here. Not so much in the periphery. Like this one has two, one in like the center, one in the periphery. The difference is that nuclei are larger than the nuclei of skeletal muscle. So the nuclei here much larger, and sometimes you will see fibers, large fibers called Purkinje fibers, and you will see intercalated disc. So this is a nice diagram to actually give you guys. You realize no label on this? Look here, nothing. This is what would this be? This looks like the nucleus, right? Look like intercalated disc right here. Look at this. See the crossbar?
Oh, okay.
I'll tell you what
crossbar is it disc?
Yes, I'll tell you what intercalated disc is. Like, I'll show you a little bit more on it. Because the cells are long-lived, with age advancing, they accumulate a pigment called lipofuscin. Remember, we said that the cardiac myocytes don't have the satellite cells. They don't regenerate. They accumulate a lipid structure in the cell. But it's highly vascularized because you need a lot of blood vessels and a lot of capillaries to supply the heart. Even though the heart is pumping all the blood, or 5 liters, 5.25 liters of blood throughout your entire body, it will need to have capillaries, arteries, arterioles supplying the organ itself. So you're going to have a lot of blood vessels as well for the cardiac muscle. Here, the diagram, the arrangement is basically the same for cardiac muscle. It's just that they are branched and they're shorter. So the fibers are much shorter. The muscle cells are branched and they're shorter and they're joined. Sometimes by the end, you have the intercalated disc. That's how the cell communicates to the next cell beside it. So here, if you look on this diagram, it's showing, you expect the sacral lemma just the same. It's similar to the skeletal muscle except for the few differences that we noted. The nuclei or nucleus, if it's more than one, much bigger, and they're both striated, yes, but there's intercalated disc for this one. They have the myofilament, the actin and myosin, so they might have the alternating light and dark area just the same because they're striated. They'll have the abundance of mitochondria. There's going to be the Sacroplasmic Reticulum that is going to be important for action potential just the same. Remember, the cardiac muscle is an involuntary muscle, so action potential has to be generated all the time continuously. The intercalated disc, and this I would say you need to know, intercalated disc is made up of 3 things. You already know there is a gap junction that allow the passage of ion across for action potential. There's the desmosome, there's fascia adherens, and you'll see it eventually in a different view. The cardiac muscle has loose myofilament or myofibrils. They also have closely packed mitochondria, and they have a contractile unit too, just like the skeletal muscle, the sacromere. So if you look at this diagram over here, you see a lot of mitochondria closely packed together, closely packed mitochondria. Probably can use the imagination. It would be the same. The muscle fibers, so like the dark area would be the myosin, the light area would be the actin, and here you have tubule, I think the T tubule. This is showing the Z line. So like this is like part of a sacromere, and you have the mitochondria that is closely associated with the sacroplasmic reticulum. And see here, and it's also closely associated with the T tubule, as you already seen in the diagram. So this is an up close look at the intercalated disc. The intercalated disc join cardiac muscle cells where cell borders interdigitate into rectangular steps or irregular width and length, irregular at the edge, as you can see, and this would be like the end of one. This is the intercalated disc, this region here. So that's why them say it's rectangular and it's irregular, because it's like the end of the cell and it's connecting this cell to this cell. And this is mitochondria, this cell, mitochondria, this cell, and you can see the striations as well. Intercalated discs are aggregates of 3 junctional specialization. The desmosomes provide mechanical stability. The desmosomes are a form of tight junction, provide mechanical stability. The fascia adherentes are sites of attachment and insertion of thin and thick filament. So you guys at least need to know that. And then you know that it also has the gap junction. So the intercalated disc consists of the desmosomes that allow for mechanical stability. It's like a tight junction. Fascia adherents, that is the site of attachment or insertion of the filament. Actin Myosin Filament actually bind to the fascia adherentes. If you see anything adhere, it's like adherence junction. Just like you have the desmosomes, it's like a anchoring junction because it's attaching to the filaments or the protein within the cell. This image is the electron micrograph. Here, the circle is where the desmosome is supposed to be. It should be the fascia adherent. This is the end of the cell, and this is the intercalated disc. So the cross wall that you see in the histology, when you saw it, you're looking at the intercalated disc. So it's fascia adherents. Remember, this is electron micrograph, so you magnify it plus. You're seeing it much bigger, which is why you can see mitochondria. Otherwise, you couldn't see a mitochondria on the histology like that. So the fascia adherents is right here. The desmosome is right here. And this is a mitochondria, as I mentioned. And this would be the rectangle, which would be the gap junction. Fascia adherents, desmosome, gap junction. Fascia adherents, desmosome, gap junction. And once you know that, you know this. The adherents, we say attached to the microfilaments, right? Actin and myosin filaments, we said that. Here are supposed to be, so the intercalated disc, these are supposed to be the desmosomes. And it's similar to the skeletal muscle, as we say, the T-tubule, Sacral Lemma, invaginated T-tubule, the myofilaments, actin, myosin filament. So the sacromere is the main contractile unit of the cardiac muscle. Just like the skeletal muscle, there is an abundance of mitochondria just the same, and it makes sense because you need the mitochondria for the power, because it's a powerhouse, right? And these are the contractile proteins. You have a lot of mitochondria. There's a mitochondria make up 25% of the volume of the cell. You can have inclusion bodies, glycogen and lipid droplets in the sacroplasm, and they're there for energy. So they're there for a natural source of energy. So if you're looking at a histology slide and you're seeing the light area, for example, on our cardiac muscle, it's glycogen droplet. Let's see if we can see the intercalated disc right here. Intercalated disc, yes, branching, glycogen. Look at this. This one has a lot more intercalated disc. And you remember what the 3 components of the intercalated disc are now?
Said desmosomes, the fascia adherentes,
Yes.
and the insertion point. I don't remember what that was. The gap junction. Yeah, gap junction.
Yeah, and we already know that the striation are as a result of the filaments. We already know a bit on the function of the intercalated disc. They're there for electrical and mechanical activity. This is just another way of looking at the intercalated disc. You have the desmosome here, you have the gap junction here, you have the fascia adherents as well. This is also looking at the gap junction.
Also, yes. So the squid is the intercalated disc.
Yes. So the intercalated disc has 3 main functions. Cellular adhesion, which part is for cellular adhesion? One name fascia adherents, one name desmosome. Electrical ionic cell coupling.
That would be the gap junction.
You know that this is the same as the skeletal muscle, but in terms of the whole excitation, contraction, coupling, contraction is a little bit more different in the cardiac muscle. There are different cells that are there for specific function in the cardiac muscle, Purkinje fibers and all of these. Sacroplasmic reticulum, as I said, there are tubular stuff, and the tubules going to interact with the sacroplasmic reticulum. Actin and myosin myosin filaments are the same, as I said, for the cardiac muscle. And as you can see, the proteins are there. And specific for the cardiac muscle, we measure these objectively to see if a person has like heart attack. The important thing is that the atrial myocytes are some cells that specialize in the synthesis and they secrete hormone. These are some specialized cells that are in the atria. Remember, the heart is divided into atrium, ventricle. Atrial cells, there are some cells in the atria that produce hormone called natriuretic peptides, and these cells are very important in helping patients who have heart problems. These peptides are released when a person is going through congestive cardiac failure to help them to excrete sodium, because in congestive cardiac failure you have pooling of the blood, right? So the heart is not pumping properly, so there's pooling of the blood, so you have water retention. So this peptide that is released from these cells will promote sodium excretion, and once you excrete sodium, you excrete water, because remember that sodium is in the extracellular compartment mostly. So these Purkinje fibers are modified cardiac muscle cells. So there are some types of cardiac muscle cells too that are scattered along the innermost part of the mitochondrium and the adjacent part of the endocardium. So they're in the innermost part of the myocardium that's in the cardiac muscle. This is just a slide showing the Purkinje fibers, and it's showing the myocardium. Important thing is that there's also glycogen here, and associated here you can see the glycogen in the Purkinje fiber. But the clinical point for this is that the patients can have sudden cardiac death, and as a result the heart stops beating, and then they have what we call ventricular fibrillation, where the ventricles beat really fast. And this ventricular fibrillation is said to be due to the enhanced or increased automaticity, and is as a result of the Purkinje fiber. This is just an ECG. So when the patient comes in, this is what we see on the ECG. So you guys should be able to relate electrical activity and producing this in a machine. There's also dilation of the heart, where the heart can be dilated, and it's usually structural change in the cardiac myocytes itself. Can be a familial thing where your heart just dilated, and obviously that predisposes you to heart disease or even heart failure. You can have also hypertrophic cardiomyopathy, where the heart is enlarged, and this can be as a result of mutation of gene encoding proteins in sacromeres. These affect like the protein binding region. Patients with high blood pressure can get hypertrophic cardiomyopathy. So the smooth muscle is the last part. It's not a lot, it's not as detailed. Main functions for emptying the bladder, peristalsis, emptying the bladder through peristalsis, as well as emptying the bowel, and pupillary constriction thing. Ryan had asked me about the smooth muscle. Erector pili muscles are smooth muscle, and as we know, it's automatic, and the uterus is an important smooth muscle. Doesn't have striation, and the walls are hollow tubules. So the smooth muscle, we know they're everywhere. They're in the digestive, respiratory, reproductive system, and this is just showing the arrangement of the muscle fibers. It can be this way, inner circular, outer longitudinal, depending on the organ. They are mononucleated, and the nucleus can be found in the widest part of the cell. When you guys have time, you can just look at this image to get the details from it. They don't have striation, and they don't have sacromere, so that eliminates most of your problem. What they do have are the filaments. So actin myosin are going to be there because they're contractile protein, and they're necessary for contraction, because remember that the smooth muscle have to contract as well, with the bowel, peristalsis, all of that is contraction. There's actin myosin filaments, and this is just showing the arrangement of the fibers and the nuclei. And the clinical point is that you can have pulmonary hypertension, and remember that the vasculatures are lined by smooth muscle. Hypertension is basically when you have narrowing of the lumen, and this is due to thickening, and there's also gene associated with this. Last thing, there is asthma, hypertension. These are all problems of the smooth muscle, and that's it. So if you guys don't have any questions, that should be it.
Questions from me.
All right. Sorry to keep you guys so long. This is probably one of the biggest lectures you'll have.
Thank you so much, Dr. Lee.
No problem. Bye.
Bye.
Thanks, you too.